Okay, this is part 2 of special topic number 3, talking about herbicides and warfare defoliants. Okay, before we get into the defoliants in Vietnam, I wanted to mention Roundup. This is herbicide that's in the news a lot. It's a very safe herbicide, much like pyrethrins. Virtually non-toxic, as you can see, very high LD50, meaning low toxicity. It's about half as toxic as table salt, as you can see. Soluble in water. It's not carcinogenic, not mutagenic. It really does a very good job at killing plants because it interacts with an enzyme found in plants and not found in mammals or animals per se. And again, here's the toxicity of Roundup, as you can see, compared to some other things. Okay, this is actually one half of Agent Orange 2,4-D, and this stands for 2,4-dichlorophenoxyacetic acid. So 2,4-dichlorophenoxy, the phenol group, acetic acid. Fairly non-toxic, as you can see, LD50. It undergoes metabolism in the soil, half-life of about 7 days, depends on the pH and the microbes in the soil, used on food crops, really very safe. And the important thing is, as you're going to see, it's not the same as 2,4,5-T, even though the lay press and politicians, regulators tend to confuse 2,4-D with 2,4,5-T. They're totally different by the way they're synthesized, even though they differ by just one chlorine, as you'll see. And here's the degradation of 2,4-D. Again, you have an electron-rich benzene ring, despite the chlorines. It's metabolized and eventually broken down. Once you have 2 hydroxyl groups on the ring, then the ring is easy to break down because it's so electron-rich. And in fact, oxidation is the root of metabolism for most things in us and in plants. And when you have 2 OHs, readily oxidized, breaks down. And once you break the benzene ring, then of course it's predestined to go rapidly down to acetic acid, carbon dioxide, sodium chloride in the soil, once you break the aromaticity of the ring. Okay, in Vietnam, the war that began in the late 60s, mid-60s or so, used, as you'll see, the purpose of these defoliants, these herbicides. Agent Orange was the major one. That was a mixture of 2,4,5-T and 2,4-D as the butyl esters. But the active compounds in both cases are the phenoxyacetic acids. The ester group is hydrolyzed in the environment, and it's actually what kills the plants are the phenoxyacetic acids. Agent White was also used. That's a mixture of 2,4-D and picloram, which is a different herbicide we're not going to talk about. Agent Blue was an arsenic compound. This was used to kill the rice crops in South Vietnam, the Mekong Delta, so-called. We're not going to talk about these at all, but just focus on Agent Orange. Named after the color of the drums, and you can see from 62 to 69, about 4 million acres was defoliated. This is roughly the size of Connecticut, Rhode Island, and I think Massachusetts combined, a very large area. Vietnam is not a big country. And this amounted to some 88 million pounds, or 50 million pounds of 2,4,5-T. Now dioxin we're going to talk a lot about. I'm going to put the structure of dioxin up now, just so you see what it is. It's actually the prototype of a series of compounds, but this is the one that caused all the problems, at least initially. It's called dioxin. It's a misnomer because, as you'll see, dioxin itself has never been synthesized. Dioxin is this part of the molecule, but this is TCDD, stands for tetrachloro-dibenzodioxin. And when the war was over in Vietnam, they analyzed the surplus examples or samples of Agent Orange, and they found that most of the samples contained less than half a part per million of this compound. 4% of the samples contain greater than 10 parts per million. Still a very small number, as you're going to see, but that number, 10 parts per million, was enough to cause birth defects in animals and perhaps in man, and certainly would cause chloracne. Average concentration about 2 parts per million. One sample had very high levels, as these things go, 47 parts per million. Again, it was used basically to deny cover to the enemy, the Viet Cong and North Vietnamese. Did a very good job, as you can see. Just total defoliation of parts of the jungle in Vietnam. Comparable to the gypsy moth caterpillar, as you can see. Okay, let's go back in time. It turns out in 1899, again in Germany, the Germans were the best chemists in the world in that time period. And this guy Herxheimer discovered chloracne, which is a skin disease much like teenage acne, blackheads and scarring and pus, terrible disease. And he thought it was due to chlorine gas, because it was noticed in the workers making chlorine from actually table salt. But then in 1957, again in Germany, these guys were able to isolate this compound on the blackboard, TCDD, from 2,4,5-trichlorophenol, and demonstrated that compound caused the chloracne, not the 2,4,5-trichlorophenol or phenol itself, but rather this compound. Now, this paper was written in German, of course. Nobody saw this paper in this country, apparently. They didn't realize that, in fact, what was causing the problems that we're going to see later was, in fact, this compound and not the 2,4,5-T, the trichlorophenol or the phenoxyacetic acid. This study was widely noted, and these guys noticed birth defects in mice, other animals, given 2,4,5-trichlorophenoxyacetic acid, or Agent Orange, basically. They didn't realize at the time that their sample they used in this study was heavily contaminated with dioxin. If they had read the paper in '57, if they'd known about that, they would have tested for this, but it was unknown at the time. And this paper received a lot of publicity, and it was assumed that then that Agent Orange itself, 2,4,5-T, was responsible for the birth defects that these guys noticed in animals. You can ignore this one. And then in 1970 at Dow, and Dow was one of the main producers of Agent Orange, noticed that pure dioxin caused these birth defects at very low levels in animals. Now, these birth defects are really severe, as you'll see in a minute, but chloracne, which goes way back, again, a very serious skin disease, it can persist for years, eventually clearing up in most cases. And you may recall that the president of the Ukraine, Yushchenko, was poisoned at a state dinner more than 10 years ago. Your parents might recall this. He was given apparently dioxin in his soup by the KGB, and this is how he looked before exposure, and you can see what happened. Very severe scarring. Today, he's almost totally recovered. But he was given, they thought, a lethal dose, but he's fine now. No cancer, no kidney damage, no long-term effects as far as we can tell. This happened in 2004. And he was given, apparently, a pure sample of this. So it was clearly synthesized, and then that's the most toxic conjugate, as you'll see, isomer in this category. Got very sick right away. Blood level, you can see very, very high. Our blood level is less than 0.02 parts per billion if we have any in us, and we probably have some. He's probably given about 1 milligram, maybe a little bit more than that in his soup, which turns out to be about the lethal dose in monkeys, but not in man, as it turns out. These birth defects are really hideous. Lack of eyes, lack of heads in the animals. I mean, that's a serious birth defect, obviously. And it's 1 million times more teratogenic, that is, birth-causing effects, than thalidomide, which was a sedative given to pregnant women in the UK in the 50s. And this caused just unbelievable birth defects in the offspring of these kids. This is a thalidomide. Ironically, an isomer of that, because that has a chiral center, is used to treat leprosy, the isomer of thalidomide. But you can see it caused massive birth defects. But dioxin's 1,000,000 times more teratogenic in terms of a dose. Here is a normal rat fetus. And here's a rat fetus where its mother was given 2 parts per billion. Micrograms per kilogram corresponds to 2 parts per billion, if you think about it. Serious birth defects, obviously. And this is a very small amount, very small dose. In 1974, I wrote a paper on this because it looked like dioxin was going to be the doomsday chemical of the 20th century, based on the toxicity, as you just saw, and also in guinea pigs. It kills guinea pigs at very, very low levels, parts per trillion almost. So I wrote this paper, again, in 1974, but now we know it's not as toxic as we once thought, as I once thought in 1974. Let me show you what a part per million is, because it really is a very small quantity. And you can devise your own analogies if you want. 1 inch in 16 miles, that's a part per million. And in 2 years. And if your parents like a dry martini, this is 1 drop of vermouth. So a very small quantity. Now, a part per billion is 1,000 times smaller. And the lay press, consumers, politicians often confuse parts per billion, parts per trillion, kind of all the same. They're not the same. A part per billion now is 1,000 times smaller. So again, analogies. This is changing all the time, of course. I actually weighed a crouton for this. Medium-sized crouton, to get this analogy. I mean, that's a huge salad with 1 little crouton. That's a part per billion. Oh, no, well, he's out of the picture, I guess. Okay, a part per trillion is 1,000 times smaller than a part per billion, 1,000,000 times smaller than a part per million. So these things are really small. And the point is, very few chemicals are toxic at levels of parts per billion, much less parts per trillion. Plutonium, Botox are probably the only 2 compounds that are toxic at this level. This is mind-boggling to me. That's a part per trillion. And lots of others, too. Okay, again, dioxin itself is this compound. This is 1,4-dioxin or paradioxin. Now, why is that unknown? That's been very difficult to synthesize. Now, this goes back to something you may have had in Chem 51, Chapter 15. Any thoughts about the aromatic character or lack thereof of the ring? Did you have anti-aromaticity at all? Okay, well, this has actually 8 pi electrons, 2 from here, 2 out here that don't count, 2 here, 2 don't count. So 8 pi electrons, anti-aromatic. So in fact, that's unstable, unknown compound. And that is dioxin. So it's a misnomer to call this dioxin. This is dibenzoparadioxin. And then here is TCDD, as you can see. This is the numbering that you should know, I think, the numbering, because we're going to talk about that separately. This is how you number it. And this is the one that is the contaminant. When you synthesize 2,4,5-T, that's what you get. If the temperature isn't controlled, that's the key. And you'll see that in just a bit. So here's a model of dioxin. It's a white solid, looks like salt or sugar, any chemical that is not colored, obviously. It's a solid. Okay, this slide's important. Now, this is how you make 2,4-D. This is chemistry you guys have all had before in this term. Phenol, as you know, is a very reactive benzene ring. It chlorinates easily, twice, orthopara. You can stop it after 3 times. You don't even need a Lewis acid catalyst. This is degrees centigrade. And now to convert this to 2,4-D, 2,4-dichlorophenoxyacetic acid, it's just an SN2 reaction. Sodium hydroxide on phenol, as we've had a bunch of times, removes the acidic proton, then an SN2 attack on chloroacetic acid as the carboxylate gives you that after you put the proton back on. Basic chemistry we've had, just like a Williamson ether synthesis. Now, 2,4,5-T is made a different way. The reason you have both, apparently, is that they're active against different kinds of plants. 2,4-D, 2,4,5-T is a perfect mix to kill all the vegetation in Vietnam, for example. If you chlorinate benzene 4 times, again, the chlorine's an orthopair director, and if you go through the exercise, this is the major product, because you don't want 3 or 4 chlorines in a row. So that's the major product. Now you have to convert this to this. And the way you do that is sodium hydroxide at high temperatures under pressure. Now, it might go by a benzyne mechanism, but you never see that isomer. So I think the mechanism is just a Meisenheimer. Addition, elimination, hydroxide attacking, all 4 chlorines are the same. And then the negative charge is stabilized inductively mainly by this chlorine and this chlorine. And then, of course, the electrons go back and kick out the chloride, and that will give you the phenoxyacetic acid. You treat that with the chloroacetic acid, and then you form the SN2 reaction here to here, and that gives you, after acidification, 2,4,5-trichlorophenoxyacetic acid. But if the temperature goes above 160, and this was not realized by all 7 chemical companies making this compound in the 60s, you get a dimerization. Again, by a double Meisenheimer. So 2 of these get together, and this one attacks this ring, and this phenoxide on the second one attacks this ring, and you get this. It's not a very efficient reaction. But again, because it's so toxic, relative to this, 2,4,5-T, it is a problem if the temperature is allowed to go too high. You can see at 300 degrees, the yield is only 10%, so it's not a very good reaction. But at temperatures above 160, you're still getting this dimerization to some extent. Okay, this compound was produced by all these companies for use in Vietnam, and they used a lot of it, millions of pounds of 2,4,5-T during that 7-year period. One of these companies had very sloppy temperature control, as you'll see, because they analyzed the 2,4,5-T samples after the war, early 1970s or so. 19 samples were pretty clean, maybe 0 or at least less than 0.1 parts per million. 7 samples between 0.5 and 10 parts per million, and 13 samples greater than 10 parts per million. 15 of those samples with relatively high concentrations of dioxin were from one chemical company, and I don't know which one. One of these guys had sloppy temperature control, and that led to this contaminated Agent Orange. Yeah?
I have a question. When you say manufacturers of 2,4,5-T and Agent Orange, is Agent Orange therefore not 2,4,5-T? It's just the 2,4-D?
No, it's a 50-50 mix of both.
Of both.
Okay. Yeah, that should be on one of the first slides.
Yeah. So it's
Agent Orange
and Agent Orange, same thing.
Same thing, yeah. Yeah, again, it's half and half. And then 2,4-D does other things. It's not toxic, but it's always there by definition. I mean, they mixed them together and then sprayed that mixture. It was used in this country along with 2,4-D, railroad tracks, farms, just to control weeds and brush. It does a very good job, as 2,4-D does. You can buy 2,4-D now, of course, and use that. It's very safe, very effective at killing weeds, dandelions, for example. The properties, as you might guess, this thing, because it has a lot of chlorines on the ring is relatively stable. It's planar, high melting point, as you can see, insoluble in water. So again, biomagnification, as we saw with DDT. stable to chemical reagents. I mean, concentrated hydroxide, concentrated sulfuric acid do nothing to dioxin. This is the temperature of the Mount St. Helens eruption in 1980. It doesn't destroy the dioxin. Sunlight, though, if you can dissolve the dioxin, sunlight, as you'll learn about if you take Chem 63, has the ability to break the carbon-chlorine and carbon-bromine bond. And if you take off the chlorines, you have a safe compound. It's metabolized in the soil slowly, as you can see. It doesn't get into plants. If it's in the soil, it tends to stay there until it's slowly hydroxylated and metabolized by bugs. It doesn't get into crops, for example. Very, very toxic. Again, here's LD50 in milligrams per kilogram. Again, this corresponds to parts per million, but it's confusing if you think about it like that. Again, it's milligrams of compound per kilogram of animal body weight that will kill half of the animals that you test. LD50, lethal dose. You can see the lower the number, the more toxic the compound, and only cobra venom and botulinus toxin are more toxic acutely than dioxin. 2,4,5-T, 2,4-D, not too bad. Nerve gas, all of these things we've talked about a little bit, but you can see it's only less toxic than cobra venom and Botox. Okay, different animals, you can see the toxicity. To my knowledge, no one's been killed, even though people have been exposed to dioxin. As you'll see, no one has apparently died from it, despite what you might read. And one of the first animals to be tested was the guinea pig. And the guinea pig, when I wrote my paper in 1974, that was what people were looking at. Guinea pigs die at 0.6 parts per billion. It's enough to kill a guinea pig. I mean, that was amazing. But these other animals are not nearly as susceptible as the guinea pig. What about chronic toxicity? You know, this is something you have to worry about, obviously. Cancer, kidney disease, diabetes. Here's a study in rats where they were given a small amount. This is 0.1 parts per billion per day for 2 years. Every day in their food or water, they were given this. And indeed, they had increased liver and lung cancer, but decreased, these tumors actually decreased for some reason, and increased mortality. So now in another group, they decreased the levels by a factor of 10. So now they're given, as you can see, this corresponds to 10 parts per trillion per day, very small amounts. So 7 parts per billion total over 2 years, very small amounts. Still, you have liver damage. Mortality, though, seemed to be about the same. Decrease it by another factor of 10, and this still corresponds to a lot of molecules, and we'll come back to that later, but no effects at these very low levels. And I show you this because back in the 70s, some scientists actually believed that a single molecule of dioxin would express toxicity. And that's clearly not the case, because you're still talking about thousands and probably millions of molecules at that low level, as you'll see. Molecules are really small, and you could do the calculation yourself. Importantly, pure 2,4,5-T, the trichlorophenol or the phenoxyacetic acid, no toxic effects at these massive amounts compared to these guys in terms of chronic toxicity. Okay, this is a case in 1971 where oil was sprayed in a horse arena to control the dust, and the waste oil was obtained, as you'll see, from a plant making 2,4,5-T, high temperature, no control, and the oil was contaminated with 32 parts per million of dioxin, a very high level as these things go. And horses died. Horses turn out to be very susceptible to small amounts of dioxin. As you can see, children who played in this arena, who lived on the farm, actually developed chloracne, and that's the hallmark of exposure to dioxin, is the chloracne. they traced this oil to a plant that made hexachlorophene. In fact, this is talked about in McMurray, one of the problems in chapter 16, I guess, or 17. It talks about this. And hexachlorophene is made from 2,4,5-trichlorophenol. And if the 2,4,5-trichlorophenol is not carefully made with regard to temperature, you're going to get dioxin. And that was the case in this arena. Here's the synthesis. Again, this is a problem in chapter 16. 16, not 17, yeah. Where this reaction is similar to a Friedel-Crafts alkylation. If you take an aldehyde like formaldehyde, in strong acid, you protonate it, and you basically make a carbocation. And then Friedel-Crafts alkylation, this is the only position because other ortho para positions are blocked. And then you get an alcohol here, CH2OH, if you think about that. So a second molecule, number 2 of this, attacks the carbocation that you'll get when the alcohol is protonated. And so you get this compound, the diphenyl methane. And that was used as an antiseptic for years in hospitals. In fact, newborn babies were lathered with Physohex. And in many cases, I'm sure, this sample was contaminated with dioxin. Nature, of course, makes very similar things. Here's a compound isolated from a green seaweed. You can see it's a brominated, hydroxylated diphenyl methane. And again, you can recognize a natural compound because of the OHs. The phenols always, or the diphenyl ethers, diphenyl methanes, in addition to halogen, will always have OHs on the ring. This is no longer used, as far as I know. Here's another compound that's related. This has been in the news a lot. And again, nature makes a complement, similar compounds that we've talked about before. Okay, let me summarize dioxin toxicity. Very low levels will cause cancer in animals. Man, 9 parts per billion is enough to trigger chloracne in man. And birth defects in chickens, very low levels. Okay, now, we're not over yet. The amazing part of this is that if you take off, let's just take off this one and this one, that compound is less toxic than table salt. You abolish totally the toxicity that you see at levels of parts per billion for the tetrachloro compound. Let me show you that. Okay, these are 2 compounds that are very toxic. The ones in red, highly toxic. These guys are essentially non-toxic. You can't give a rat enough of this compound to kill it. It might choke to death, but it is impervious to the toxicity of these compounds. Very interesting case. This is the important slide that describes that. In order to be toxic, a dioxin molecule has to have at least 3 chlorines or bromines, as it turns out, at the so-called lateral positions, the side positions, these positions, and 1 open hydrogen, that is 1 open position at a peri position, and that corresponds to this position. So here, those are peri positions. And if you satisfy those criteria, then in fact you'll have a toxic dioxin. And it can be chlorines, bromines, mixtures of chlorines and bromines, probably fluorines as well. So these compounds are very toxic. Again, you have 4 lateral chlorines here, 1 open peri position. Here, no peri position, open, non-toxic. Here, in fact, you only have 2 lateral chlorines, non-toxic. In addition, you can have a different molecule that has about the same shape. This is called an isostere, the same shape as dioxin. So a naphthalene compound that has halogens, bromines or chlorines, at these side positions, even though it's a little bit shorter lengthwise than dioxin, still is toxic and will interact with what's called an AH receptor, a protein in all of us. Furan is a compound we talked about before with regard to trapping benzene, you may recall. This is dibenzofuran. Also toxic if you have at least 3 chlorines at these lateral positions and 1 open hydrogen. Some more compounds. You can have this compound. We encountered this. In fact, this was on the last exam. If you generate, without the chlorines, you generate benzene and you don't trap it, it dimerizes, right, to form the 4-membered ring. If you have chlorines here, it's much like TCDD and is toxic. And again, here's the dibenzofuran, naphthalines. This is an interesting case where this biphenyl with chlorines here is toxic, like dioxin. This one is not. It's in blue, you can tell it's not toxic. Now why is that, in fact? Because we did talk about biphenyls before, the first week of class. They tend to be twisted. And if it's twisted with orthochlorines, it can never be coplanar. To be toxic, the molecule, the dioxin, the dibenzofuran, the naphthalene has to be planar. And that gets into this protein, the AH receptor. So biphenyls that have chlorines or bromines here can never be planar because of a steric effect. But if the chlorines are out here, it can be planar, and in fact, it's as toxic as dioxin. Okay, even more surprising is the fact that dioxin is a natural product. I mean, this really blew away environmental radicals and politicians, regulators as well. Turns out when you burn wood, wood contains carbon and chloride. They conspire to form lots of different things, methyl chloride and some dioxins. And you can demonstrate this in the fireplace as well. There are lots of forest fires around the world, and dioxins are forming to some extent. In agreement with this, if you look at ancient soil samples that have been archived long before phenols were synthesized by man, you find dioxins. And I'll get back to this. The graduate student who first made dioxins in 1957, working with our old friend Gilman, almost forgot his name, suffered from chloracne terribly. He made massive amounts of dioxins for another purpose, and they didn't know about the study in Germany, which was also 1957. And Gilman never reported this. Had he reported this to somebody, then the fiasco in Vietnam might have been avoided. And I'll show you what he did in just a minute. Yushchenko we talked about. Ironically, if you have a dioxin with chlorines at 1, 3, 6, and 8, so 1, 3, 6, if you have that compound, that's active against breast cancer. It's not predicted. You guys would now predict, based on what we've been talking about, this would not be toxic because it only has 2 chlorines at lateral positions. And in fact, it has activity against certain kinds of cancer. Don't worry about that. Okay, this is a letter I got from Dietrich, the guy who synthesized dioxins in Gilman's laboratory. What Gilman was doing was he was making things like bromobenzene and chlorobenzene and converting them to the lithiated compounds, like the Grignards, and then looking at chemistry of those. So he made these compounds. He noted, in fact, that the brominated compounds, even more toxic. Severe chloracne, he had to be hospitalized. He made grams amount of both dioxins. Got very sick, but he's alive today. At least a few years ago, he was alive. And this is what he did. He made the parent dioxin just by this coupling. Don't have to know this chemistry really. And then if you brominate this, it tends to go para out to one of the oxygens. By symmetry, they're all the same, rather than ortho, steric repulsion by the oxygens maybe. So then if you brominate this one, you guys would now predict it should go in this ring, right? The right-hand ring is deactivated by the bromine. It goes here, and then finally, if you brominate it 4 times, 8 equivalents, excess, you actually get this compound. And that, of course, is extremely toxic, more toxic than the TCDD on the board. But he never reported this, even though the guy was really sick. He was hospitalized for months with terrible chloracne and a lot of liver dysfunction, I think, as well. Okay, there's another case in Austria where it seems to be a deliberate poisoning act where 2 women actually were exposed to very high levels of dioxin. And as far as I know, they're both alive today, but severe chloracne, as you can see. And what was found in this study is that olestra, this fat substitute that's not talked about much anymore, has the ability to extract the dioxin from your fat and remove it. And that's currently the only treatment for getting rid of dioxin in your tissues. Okay, dioxin is formed when you burn things. Cigarette smoke has the octachloro. Now the octachloro is non-toxic. Hexachloro is. Hexachloro might be this one. And you guys would now predict that would be toxic, right? And is. Diesel exhaust as well includes even some of the tetrachloro. That's the prototype. So it really is everywhere. Small amounts. These are parts per billion. Big city dust. And around the Dow chemical plant, of course, they were making a lot of Agent Orange. Way back in 1949, there were accidents in the manufacture of the 2,4,5-trichlorophenol because that is in a bomb, high temperature, high pressure, and oftentimes you get explosions. And you can see the workers develop chloracne. But no cancer and mortality's about the same. Dow, of course, made a lot of this compound, no chloracne, another case. And they were concerned that the wives of workers exposed to dioxin might develop birth defects. And that, at least, did not seem to be the case. Then in Italy in 1976, again, giant explosion, the worst one yet, I think, up to this point, in Seveso, Italy, where all the birds and animals died in the field around this plant. Kids playing outside, and also adults, developed chloracne, severe chloracne. And because they knew about the birth defect study in 1969, a lot of women elected to have abortions. And unfortunately, all of the fetuses, I think, were normal, as it turns out, even though they elected. They were so concerned about the birth defects that you saw in rats. And cancer is still not a problem, apparently, at least as of about 2010. Here's more on that Italian accident. Again, they still have chloracne, some of these kids that had very large doses, as you can see. Okay, this slide again illustrates the fact that molecules are really small, and it's not reasonable to think that a single molecule of anything will be toxic. It turns out we can detect, using GC or HPLC, 1 part per trillion, and that corresponds to 180 billion molecules of dioxin. But if you go down it by a factor of 10 in a glass of milk at this level, you can't see this. A part per quintillion, you can't see the 180,000 molecules of dioxin in a single glass of milk. You'd have to get a lot of milk and concentrate it down in order to see the dioxin. So molecules are really small, but they're essentially invisible at that level in a glass of milk. Okay, I mentioned that in a forest fire you can form dioxins, and again, this has been demonstrated quite a bit. And you can imagine lots of forest fires annually, hundreds of thousands, mostly lightning induced. So dioxins are being formed, we think, all the time to some extent. Depends on the temperature, and if the vegetation is wet, then you'll tend to get more dioxin. Incomplete combustion, of course, will lead to more dioxin. In addition to that, we know that enzymes, peroxidase enzymes, have the ability to dimerize things like the chlorinated phenols. If you take this compound and incubate it with this peroxidase, you tend to get not only the dioxins, but also the dibenzofurans. Garden compost, if your parents have a compost file, and if there are chlorophenols there that you get when you break down vegetation, normally humic acid, you can, in fact, form dioxins from the peroxidase enzymes that are also there in the soil. Sewage as well, and again, forest fires and wood. Some more evidence for that. You can see here's an old sediment that was archived, thought to be 8,000 years old, found to contain some dioxins. Peter Silk, a friend of mine in New Brunswick, Canada, likes to investigate, for some reason, investigate the chemicals in peat bogs in this part of eastern Canada, and he found dioxins and chloroform there. He found these guys. This is the one that's active against cancer. And what he did, he incubated the peat with radioactively labeled chloride, went away, came back, and then isolated the dioxins and found that Cl37 was incorporated into the dioxins, indicating that it's being biosynthesized in the soil, in the peat, and also isolated the chlorophenols as well, and also the dibenzofurans. The same kind of study was done in Sweden, where this guy again took Cl37 and put it in the soil in a forest, came back a year later and isolated the dioxins that contain, again, the radioactively labeled chloride, and also the chlorophenols. So really direct evidence that nature has the ability, not only in forest fires, but under the action of enzymes, to make these dioxins. And this was really a shock to people working in the field. And so likewise, you can look around the world, and this has been done, you can see all these countries looking at old soils and clays that have been dated back more than 100 years, and you form dioxins. Not a huge amount, but they're always seem to be there. This, I think, is a part per billion. You guys could figure that out. Maybe a part per trillion. So in this country, these old lake beds, clays, contain these things. Different isomers, not always the bad ones, as you can see. And the octachloro, where you have chlorines at all these positions. Let me get rid of this toxic one, make it non-toxic. This one, of course, non-toxic, because you need at least one perihydrogen to be toxic. And that turns out to be the major isomer. And these clays are actually used for things in ceramics, as you can see. And the mechanism, you can do this in the laboratory. In fact, you can take pentachlorophenol, oxidize it, and you get eventually the octachloro-dibenzodioxin. Geophagy is a practice in Africa, parts of the world, of eating clay And of course, they're being exposed to small amounts of dioxin. Again, this is one of the toxic ones. I think 1, 2, 3. Yeah. Okay, another big surprise was that the ocean contains these things. Now, the first week of class or so, we talked a lot about the brominated diphenyl ethers, right? Sponge metabolites. I showed you the sponge that I collected that has these compounds. And I thought it was kind of only a matter of time before the people doing this work would find a bond formation here, would form cyclization. And indeed, 3 separate studies have now shown that. These brominated dioxins that occur in sponges. Now let me ask you this, what would you predict about this? 3 lateral bromines and 1 perihydrogen, you would predict to be toxic, but in fact that's not toxic. Now why is that, do you think? Well, we know that metabolism in the dioxins, the ones in Yushchenko, or 1 Yushchenko, undergoes metabolism to that, and that's excreted in the urine. And so, in fact, if your dioxin has an OH already on the ring, it is not very toxic. It's excreted in the feces or in the urine, metabolized, or connected maybe to a sugar, a glucose, or an amino acid, and it goes out. So these things, even though they satisfy the criteria we just talked about, turn out not to be very toxic. And here's the sponge that makes these things. Studies done by the USDA, in fact, found that if you take this compound and feed it to rats, the rat has the ability to cyclize this, as you can see. Or you can just take the phenol. If you don't want to make the diphenyl ether, just take pentachlorophenol, give it to cows, isolate their feces, extract, you form the dioxin. So this now begs the question, because we have these enzymes in us, as I said before, they're in our white blood cells to fight infection and bacteria. Myeloperoxidase, does that enzyme have the ability to form dioxins from the chlorophenols, some of which are found in our diet, and they're all found in the environment? And of course, you know what the answer is going to be. It does. So it seems in this, it's really gone full circle if you think about it. Now, a human biosynthesis of dioxins is certainly feasible because we have myeloperoxidase in us. If we're exposed to these chlorophenols, you might get dioxins. Now this one, not be toxic. This one depends where these chlorines are. It might be toxic, might not. If the chlorine is here and here, and one, no, it would not be toxic, no perihydrogens. This one I think would have to be toxic. But you guys can play with those.